In this lecture, there will be some concepts that are new for most of you, and hopefully there are some concepts that are a bit of a review. But whichever it is, whether you're coming to these for the first time or whether you've learned them before, it's an important refresher, and these are really critical concepts for what we're going to be discussing throughout much of the semester. So I'm going to go back to a statement that we talked about in a previous lecture by Haeckel. So if you remember, Haeckel was an artist and a biologist. He drew many beautiful pictures. I gave you a link to some of those if you're interested. And he was interested in animal form and kind of the aesthetics and the beauty of that animal form. But he was also an embryologist. And one of his main contributions was this idea of ontogeny recapitulating phylogeny. So just a refresher, ontogeny, of course, is embryological development. So think about that first 1/3 of the human gestation period. And other animals, it's also approximately 1/3 to 1/2 of that early period of time of gestation, where we go from a single fertilized cell all the way to a complete organism. There's often some growth and some post-birth and post-hatching development that occurs, but those are fairly minor compared to the major changes that occur during that ontological period. So that's ontogeny. And of course, phylogeny refers to the evolutionary connections, the history, the shared tree of life, if you will, between all organisms. And so Haeckel's idea was that this ontological progression that we saw occurring from single cell up to fully formed organism went through a same or at least a similar series of steps as the evolutionary history. Now, for a period of time in the 20th century, that idea was rejected and was pretty much kind of laughed at as kind of overly simplistic. In the 70s, Stephen Jay Gould published a book called Ontogeny and Phylogeny, where he basically kind of brought back this idea and pointed out that although, taken at 100% face value, it didn't hold water, but there were many portions of it that did. And so Gould emphasize this idea that much of evolution, much of the changes that we see between species are changes that impact the early developmental period. Those are the most dramatic and maybe the most efficacious if we're looking at something that's going to make a change between one species and the next. If we look at a change in an early developmental process, those can be major and have huge implications as the differences between species. So I'm not going to have you read any of this work, but it was a very meaningful and important work, and Stephen Jay Gould basically brought back this idea. And so now we are going to look at some of the concepts underlying this idea and how evolutionary changes, mutations that lead to differences in the ontological process can lead to massive differences between species. Now, to understand this, there's one really first kind of key idea, and this is idea of heterochrony. So this is nice Greek root words. You're familiar with them in other usages, so it should be easy for you to remember. Hetero, of course, means different or a change, and chrony, and same as chronological, refers to time. So this is a difference in the timing of development. And small changes to the timing and development can lead to massive downstream impact shifts in the way that organisms look. So we're going to look at 3 of these kind of key concepts. Now, on these diagrams, we've got a very simple 2-axis graph here. The x-axis here represents time, and the y-axis is shape. You could also think of it of size or some change, if you will, in the developmental process. Okay, and in this one, the dotted line represents kind of the normal developmental process where we begin at one period and slowly change over time if we're looking at the change in shape or the growth of a structure until we reach the final form for the way that structure is going to grow. So if you want, you could think about this as the growth of an appendage like an arm. We begin with a small cluster of cells with very little differentiation. It grows out, differentiates, and eventually we get a fully formed arm. Okay, so with that setup, let's look at how minor modifications might change the eventual outcome of this developmental process. So the first one is we are going to look at either an acceleration or a deceleration of the process. So the amount of time that elapses remains unchanged. But if the rate at which growth occurs is slowed down, and the slope of this line, of course, would represent the rate of growth of the arm, that if that is slowed down, but we still truncate development at the same time period, there's a finite window of time over which development can take place, then we're going to have a less formed, or at least a smaller, but certainly a change in the final outcome of that structure. Vice versa, of course, if we were to speed up development and given the same amount of time, we could end up with a more fully developed, larger, maybe structures would be different because there'd be more time for, say, finger differentiation or something like that. So we could have dramatic changes by simply one little change in the rate at which that process is occurring. Okay, so that's one example of heterochrony. The other examples here we're going to call hypo, meaning underdeveloped, or hypermorphosis, so either under or overdevelopment. And in this one, the rate of development remains unchanged, but the time period is changed. So if we have less time, as illustrated here in the example of hypomorphism, we're going to have a reduction in the final form of that structure. If, however, we have an extended period of time for development, then we will end up with a larger, more fully formed, whatever, again, developmental process we're looking at. We're going to have a change. And again, that's just a simple change in how much time is available for this process to occur. So whether it is the rate of that process or the amount of time, we end up with a heterochronic difference in those outcomes. The final one we're going to look at is post-displacement and pre-displacement, and this just refers to when that developmental process occurs. So if we're able to begin that developmental process a little bit early and still have the same endpoint, again, we'll end up with a more fully formed or a larger or a longer period of time for development. Or if we truncate, we have a shortened period of time by beginning the process later than we normally would. So again, small mutations might change the timing of these events and lead to a very big downstream impact. Okay, all of these are heterochrony. The classic example of heterochrony is kind of an elongation of a developmental period. I'm going to bring it up here for you. In the axolotl, or sometimes more commonly called the Mexican salamander. So this is a species of salamander that spends its entire life in the water. Most salamander species go through a period of metamorphosis where they will change from their larval form into a more adult form. And in fact, the closest living relative of the axolotl is a tiger salamander, which looks like this. Very different, that's the adult form, very different from the adult axolotl. But if you look at a juvenile tiger salamander, let's see if we can bring one up. If you look at the juvenile form, it looks much more like the adult form of the axolotl. In fact, they look nearly identical. You'd have to be an expert to determine them just based their differences of morphology. All right, so that's one example of how a change in development, and basically in the axolotl, what's occurring is a hypomorphosis, a slowing down of the developmental process so that we never get to the final adult form and we never go through that metamorphic change. And so that results in a dramatic difference between these two very closely related species. There are many other examples of heterochrony, but I just want you to kind of set that idea in your head and just be aware of that. Another key idea or key concept is this idea of heterotopy. And heterotopy is a displacement in location or a change in the area in which something forms. So if we have development of an appendage in an area where appendages normally aren't grown, that can be a major mutation. Okay, here's an example in plants where typically along the stems we only have, along the main stems we only have leaves and the petioles of these leaves coming out. However, in climbing ivy, structures grow out that are very, very similar to roots, that have these kind of branching patterns that can grab onto things, and that helps this plant to climb. And there are other mutations that allow for other climbing adaptations, such as tendrils or rhizomes or other things. But in this one, it is essentially a movement of root-like structures to a stem. And that results in a dramatic difference between ivy and its closest relatives that evolved prior to this change. Another really good example of heterotopy is the appendages in arthropods, where appendages originally were only expressed in a few segments and then have been extended to other segments in some species or been lost from certain segments in other species. And those result in dramatic changes, okay? And so for now, I'm just going to throw this question out here. We're not going to address this or answer it today, but we will in later ones and give you examples. Can we extend these ideas of heterochrony and heterotopy to genes? And just to kind of give you a little bit of background, I'm not so much talking about genes in the genome, although certainly we could do that, right? We can move a gene from one chromosome to another chromosome. But the idea is, does that change in location in the genome have an impact on how that gene is used and expressed? And in many cases, the answer is no. Whether it's on chromosome 1 or chromosome 2, as long as it can be accessed, it doesn't matter. So I'm not talking so much about heterotopy in the genome sense, where is it found in the genome, but more of where is that gene used? What parts of the body is the gene expressed in during early development? And in fact, what we might discover, and we will in some cases, is that at the morphological level, a heterochrony can be traced down to an exact heterochrony at the genetic level. Certainly also with heterotopy, if we change the location of where genes are expressed during development, we're going to get structures that look like, the structure they were supposed to be, but growing in a completely novel area. Now, before we move on, I just want to make one last important, and hopefully you may already kind of have this idea in your head, but I want to emphasize this. These mutations are just like any other mutation that drastically changes morphology. So if it's a mutation in the developmental period in the timing or in the location, it's a mutation. The vast majority of mutations, particularly those that have a negative or have, let's not say negative to start off with, but let's say have a dramatic impact on the phenotype, the vast outcome of most of those mutations is a loss of fitness. And so the vast majority of these mutations, when they occur, are removed by natural selection because they don't allow the organism to do better than it had prior to the mutation. However, every once in a while, one of these random mutations that drastically changes the phenotype results in a benefit. And so we could potentially look at the salamander example of this. If you're a salamander living in an area where you need to get up onto land and complete your life cycle, find food, find shelter, find mates, whatever it is, you need to get up onto land to do that, then a mutation that prolongs your juvenile stage is going to be a really bad thing. And so they probably occur somewhat frequently, but are mostly removed by natural selection. The axolotl, however, lives in an area where it was becoming less and less advantageous for the salamander to get up on land. It lives in central Mexico in wet areas, but these wet areas are surrounded by dry areas. And so as that dry habitat became more and more of a feature of its environment, getting up out of the pond, looking for a mate, looking for food, looking for shelter just was no longer a benefit. It was better to stay in the water, even though you had to compete with juveniles and maybe compete with fish and other organisms in that aquatic environment. It was a better option for the axolotl. And so when that random mutation occurred, prolonging its juvenile form, it was selected for and eventually became a feature of the entire species. So now all members of that species carry that new mutation. So just remember, these are like any other mutation. They're not always going to be a good thing. In fact, there was an idea that was proposed a while back of a hopeful monster, meaning a small genetic change resulting in a massive morphological change. So the organism will look maybe almost like a monster, right? Like something very, very different from every other member of its species. And it's only going to be in a very rare instance that hopeful monster, which might have arisen from a tiny genetic change resulting in a large morphological change, is going to be beneficial. Most of the time it's not. We're going to get an organism that does much worse than all other members of its species, and we wouldn't see it in the long term. If we were there watching when that mutation occurred and saw those one or few individuals that carried that mutation, we might be able to describe it, but quickly it would be removed from that population by natural selection. Okay, two more key concepts. We are going to define the idea of homology, hopefully just redefine it for most of you. But homology is a similarity either in structure or in genetics, any sort of similarity that is due to a shared common ancestry. So I'm going to give you an example, but then we're going to actually talk about it, and hopefully you'll realize this is maybe not as simple and as easy an example as you would first say. So I've kind of given you some background on this, so you might have already have some ideas about it. Is the wing of a pterodactyl and the wing of a bat, so are those two structures homologous? Now, the short answer is yes. And you say, well, some of you might be a little bit surprised, and it actually turns out that, no matter what I say, if I say yes or if I say no, that I'm going to have a portion of the class that's surprised that they were thinking the opposite, which is okay because the real answer is it's complicated. So let me talk about it. We've already done this a little bit, but let's reemphasize this. Okay? Do these structures have similarity that were inherited from a common ancestor? And if they do, then that's homology. And you can very easily see that, right? You can see the point of attachment right here after the neck in the pectoral girdle. The bones have somewhat similar, although changed in down the different lineages, but still some similarities, right? They have a humerus, a radius, and an ulna, phalanges, wrist bones, digits, all of these things, although changed in some one of the species or the other, are still somewhat similar. So it's very obviously homology. However, as a wing, right, as a flying structure, these are not homologous. The wing, the flying structure, did not come from a common ancestor. However, because both wings were derived from the forelimb, there is that level of homology there. And so it's important that we don't oversimplify homology, that there's always going to be another level as we get more involved, as we look at more and more of the fine details, we'll probably see some differences. But any similarity at all that was inherited from a common ancestor is homology. Okay? And some of these are easy, right? Feathers in birds, those are all homologous. They all came from a common ancestor. All birds have feathers because that evolved in their common ancestors. Others, like wings in tetrapod vertebrates, are going to be a little bit more of a complex answer with some elements of homology and some elements of analogy. So analogy is a general term. You can refer to it as, in fact, I'm going to put another term here, convergence. That's one you should be familiar with. So let's change that, and you just add that and make sure you understand that convergence is a form of analogy. So convergence and parallelism are both forms when structures evolve similar features, but they do it independently. They didn't get them from a common ancestor. They arose independently. And there are many, many examples of this. One of the fun ones is dorsal fins, right? So fish, many species of fish, have a prominent dorsal fin that helps them to maintain their orientation as they're swimming. Whales also have a prominent dorsal fin, but that dorsal fin evolved fairly recently and separately from the dorsal fin that we see in many of the fish. So those would be analogous structures, not homologous structures. Okay, and so the elements of the forelimb that make it a wing in the pterodactyl and the bat, or the bird for that matter, the elements that make it a wing are analogous when they are compared between these distantly related species. Now, it's very critical that we define the range of species we're talking about. If I was to show you a fruit bat and a Mexican free-tailed bat and a snub-nosed bat and ask you if the wings are homologous or analogous, then it's an easier answer, right? They're homologous both as a forelimb, which they got from a much more ancient ancestor, but within those three species of bat, they're also homologous as a wing because the bat ancestor evolved a wing once and has passed that down to all living bats today. Okay, so be aware that the scope is an important consideration when we're trying to determine questions of homology versus analogy. Okay, and structures that evolve independently, either through convergence or parallelism, are analogous, not homologous. Now, I'm going to take just a minute and talk about these two terms, convergence and parallelism. If something has arisen via parallelism or via convergence, here's the distinction. Technically, parallelism is where we have identical ancestral structures that then go through identical changes to become the same thing, but it happens independently. That's a very narrow definition, but that is the strict definition of parallelism. And in fact, I think if you were able to have enough time and resources available to study all examples that are seeming parallelisms, you would actually then at the end put them into the category of convergence. with maybe the exception of single base pair mutations in the DNA, right? So for instance, if I have two species that have an adenine at a certain position in their genome, and then they separate into two, they got that adenine from a common ancestor, they've separated into separate species, and then randomly both of them have a mutation that changes that adenine into a thymine, that would be a true parallelism. And those really are maybe the only true examples of parallelism. Convergence is similar, however, there are different changes along the evolutionary pathway that lead to the similarity. So complex things like a batwing and pterodactyl wing are very obviously convergent. They have a similar overall form and a similar function, but they have arisen at that in very different times and in very different lineages. But even some things that formerly were put in the category of parallelism might, if we were able to look all the way down to the genetic level, be better classified as convergences because we would see different mutations and steps along the way. One classic example, which we'll look at the very end of the class, is a change in melanin. So many different species originated having dark fur and then mutated into lighter forms, particularly in the Arctic area. So we've got Arctic foxes, right, which have through, I think actually the entire year, I might be mistaken. I don't know if Arctic foxes go through a color change or not. I think actually maybe they do. Let's look it up here. So during the winter, they have this white form, and during the summer, they have a little bit of a lighter, but still with coloration, right? But polar bears, Arctic fox, ermine, which are a form of a weasel that have a summer form and then a white coat light form. And there are actually dozens and dozens of different species that independently have evolved this light form. And when you look, they all use the same gene to make this white color. It's one of the genes involved in the production of melanin. But when you look at the mutations, there are separate mutations that lead to the same genetic outcome. The gene still doesn't produce and sequester melanin, but it's a different mutation, different areas of the gene. And so if all we knew is, oh, look, the same gene evolved in the ermine and the fox and the bear, then we might say, oh, it's maybe homologous because it's the same gene. But when we start to investigate at a more detailed level and look at the actual changes, then we realize that, oh, yeah, so it is convergence, that it wasn't the same change at all. It's not homologous, and it's not even parallelism because different changes led to those overall similarities. Okay, so be aware of those examples. I may give you other examples, but with enough supporting details so you could recognize whether it was homologous or analogous. We're going to complete this discussion with two overviews. Now, one I'm only going to do a little bit because you had a paper that gave you a lot of the history and a little bit of the science for the Spemann and Mangold experiments. But I do want to emphasize and talk about, so you can kind of draw out from there what the important idea is. The reading questions will also give you some of the idea. If you can answer those and feel okay about answering those reading questions, you probably got the major concepts from this paper. But this goes back to the early 20th century. In the 1920s, Spemann began these experiments on amphibians. Some of the early ones were done on frogs, but most of them were completed in salamanders. And they did things like divide early embryos using a fine hair and then looking to see what happened. And what they found is they could make what they called artificial twins if they did it early enough. So if you take a very early stage embryo and divide it in half, both of those halves would go on to make fully formed organisms. And this happens naturally all the time, sometimes more commonly in some species, but it occurs in humans, right, where we have a single fertilized egg that divides during the developmental period, physically separates into two separate embryos, and both of those go on to become fully formed twins. We call those identical twins, or the technical term is monozygotic. So that wasn't a new idea, but the idea that they could do it artificially and induce twinning was kind of a breakthrough and was really kind of a dramatic thing. The second major breakthrough that they found was this idea of induction via an organizer. So we're going to define both of those terms. And this is a diagram showing some of the experiments. Originally, these experiments were done with partial separation instead of completely separating embryos. They partially separated them. Or later done via transplantation. We'll look at the transplantation experiment here in a little bit. But after many experiments, lots of reinforcement, multiple trials, Spemann and his very capable assistant, I think Ingrid was her first name, Mangold. She did her PhD under Spemann and did much of the work that led to this really breakthrough paper. Later, Spemann was given the Nobel Prize. By that time, Mangold had passed away, so was not given the Nobel Prize, and there's been some argument to whether posthumously maybe she should have been, because much of the work was hers, although the ideas and the foundational work was originally done by Spemann. So we'll just refer to them as the Spemann and Mangold and give them equal credit for this breakthrough that led to the Nobel Prize. And so the idea was that we could take a bit of one organism, a developing embryo, and graft it onto another organism and induce or cause changes. So the little group of cells, and at the time they didn't know what it was in those cells, but those cells themselves were referred to as the organizer. Okay? And that little group of cells somehow provided a signal that would induce cells in another part of the embryo to go down a different developmental pathway than they would otherwise. So originally the idea was that at maybe even at the very beginning, but certainly early on in development, that all cells were locked into certain cell fate. And there are all these, and today people are still doing this, they're developing cell fate maps where they can look at the first cell turning into 2 cells, turning into 4 cells, and map the eventual fate of every single cell in an embryo. For simple embryos like C. elegans, and to some extent, at least through the early stages in amphibians, those cell fate maps have been fairly well documented. For more complex organisms that are harder to work with, we don't have complete cell fate maps. But the idea was that cells were programmed to become something, and they were programmed maybe even from the very, very beginning, from the first time they were formed. However, this group of experiments showed that cell fate could be changed, that was not locked in until much, much later in development. And so at an early enough stage, but still not, super early, if you took a little bit of a other embryo, and the most common and most famous experiment was a transplant of the dorsal lip of the blastopore. So remember, this blastopore in these organisms is going to be forming the gastrointestinal tract. And so they would take this little bit and they would transplant it onto, say, the backside of another embryo at a similar stage. And if that occurred, then rather than a single embryo, you would end up with what was at the time called a Siamese twin, and that name's misleading, but a better term is conjoined twin. But you would make an artificial conjoined twin. Not only that, but this twin was not completely composed of the cells and cell descendants from the transplant, but cells next to it would actually be recruited. So this little organizer, this group of cells, would act as an organizing center for the cells around it and would begin to tell these cells around that, say, hey, maybe originally you were going to become the dorsal side or the ventral side, but now you're going to become the dorsal side. And so we ended up with 2 dorsal sides and then a conjoined ventral side. And so this group of cells, called an organizer, was able to change the fate of the cells around it. It was able to induce a different outcome. So those are those 2 forms. Organizer is the group of cells originally, and they had no idea what the substance in it that was creating the signal, and that it was able to cause induction, or in other words, induce the cells around it to a different cell fate. Now, 2 key points before we move on to the next thing. If these experiments are done at a later stage, then we don't get this outcome. We just get a group of cells that are signaling around it, but the cells don't essentially respond to that organizer system. So it has to be at an early enough stage before these cells get locked into their cell fate. At some point, but much later than was originally thought, we do have basically a firm identification of those cells. They're locked into whatever their cell fate is going to become, or at least into a smaller range of cell fates. So they might be locked into neurological development, but then they could become either, nerves or a supporting tissue around the nerve or the brain itself or something like this. But the farther we go on, the more and more locked in cells are. And so this leads also to the idea, immediately you might be thinking of stem cells. Now, most of our stem cells that we are using today experimentally came from very early embryos that were derived and preserved prior to this differentiation. And there's been dramatic success over the last decade or so in trying to take a cell that has been already induced, that has been locked into a cell, into a certain fate, and kind of be able to turn back the clock to make a stem cell from a cell that's already been differentiated. There's been a lot of progress on that, although there's still a lot of work to be done there. And so really the most effective and widely used stem cells even today are still from early embryos that were pre-differentiation. Okay, so that's one key concept, is that there is a cutoff, or at least a series of events that will make cells no longer inducible, okay, that they are locked into their cell fate. All right. I forget what the second thing is. If I come to it, if not, we'll hit it later on. But so just review and remember that. All right, the final bit of, I remember what it is before we move on. Let's talk about what the substance is in the organizer that actually accomplishes the induction of cells around it. Now today we realize that it's got to be some sort of a gene, or maybe more specifically, a gene product. And the gene product most commonly is the mRNAs that then are made into proteins that will regulate the expression of target genes. These are these toolkit genes that we identified in the previous lecture. And it's a rather small, usually a few hundred genes in the organism that are critical during early development. So if I take a set of cells here that are producing a certain gene and put it in an area where it's not normally supposed to be, this is an artificial heterotopy, right? If I put them in that area, then as long as they have cells around them that are still inducible, we'll get an entirely new sequence of development forming here, conflicting with the primary sequence, and that's where you end up with the conjoined twins. Okay, so just be aware of that and know that it's genes, or more specifically gene products, that are the signal the organizers are sending to the cells around them. Now, in the mid-70s and into the late 70s, Lewis was doing some work with Drosophila. So by this time, Drosophila had been established as a really important model organism, was a powerhouse, a workhorse for lots of genetic work and other work with chromosomes. But Lewis started working at him with the idea of evo-devo. And he was primarily interested in a subclass of mutations. He didn't really care so much about the mutations that changed the eyes to red or made the wings curl up or caused like unusual tumor-like growths. There are lots of different mutations. But he was interested in a small class of mutations that caused structures to jump around, a form of heterotopy. So this is a wild-type fruit fly here on the left with its antenna, kind of modified antenna in flies. They're different from most insect antenna. But that's a standard antenna for a fly. But in this homeotic mutant, they grew legs, and pretty much fully formed legs with all of the parts of a fruit fly leg, a little bit of shape differences and deformation, but primarily a fully formed leg, but where an antenna should be. And so this led to his idea of a homeotic mutation where we could have a small genetic change leading to a dramatic downstream impact. And in fact, he identified a series of genes that he called homeotic genes, or for short, Hox genes, that had this impact, where we would grow legs in place of antenna. And so he called the gene, once he was able to identify it and locate it, he called that gene antennapedia. Okay? In another experiment, they located a gene that grew wings where normally a fly only has halteres. So the ordinal name for flies is Diptera, which means two wings. Unlike most flying insects, they only have a pair of wings rather than four. And their hind wings are modified into these little sensory structures called halteres. But with a small change to a single gene, they were able to grow fully formed wings in place of the halteres. So that one gene is obviously responsible for the mutation that allows for flies to have halteres in place of wings. And so they begin to identify a subset of genes, toolkit genes, and of some very important ones at this stage of development called Hox genes. We're going to be identifying and talking about the 8 Hox genes that have been identified in fruit flies, and then talk about them in other animals, but those will take place in later structures. So just know that Lewis in the 70s began to describe these. He originally found them through these homeotic mutations and was able to trace those mutations to single genes that led to these major shifts in the way that insects, particularly the fruit fly, were built.